Welcome to ICDD's in-session webinars and Dr. Joel Reid's presentation, The Canadian Light Source, CLS, an introduction to some of our synchrotron techniques. Please remember to visit icdd.com for information on free webinars, educational events, PDF databases, and JADE software.
So my name's Joel Reid. I'm a senior scientist in the Industry Services Group at the Canadian Light Source. I'm really happy to be here with you today. So today I'm going to give you a brief introduction to synchrotrons in general, the CLS specifically, and our Industry Services Group. Then I'm going to illustrate a few of our capabilities with some short examples. There will be a variety of examples drawn from numerous different disciplines using both industry projects and some published work as well. So first things first, what is a synchrotron? A synchrotron is a particle accelerator which circulates a beam of electrons around a storage ring, with beam lines coming off of that ring tangentially. Every beam line has a magnetic element, and that's used to bend the electron beam, and it keeps the electrons going on a roughly circular path and creates an acceleration, which gives rise to synchrotron radiation. Synchrotron radiation covers a wide energy range. So individual beam lines are designed to make optimal use of different regions of the electromagnetic spectrum and to use different techniques. Ultimately, a synchrotron is a materials analysis tool with many different techniques and capabilities. Oh, sorry about that. So every synchrotron has multiple experimental end stations called beamlines, which provide a wide range of different techniques. Beamlines generally have three components. One, a primary optical hutch with optical components which are used to focus the beam and to select the wavelength or energy for your experiment. An experimental hutch where the sample and experimental apparatus and detectors are placed, and a control area for running the experiment and data acquisition. And researchers from many different disciplines travel to synchrotrons to use beamlines with specific capabilities. Here's a sampling of different areas where synchrotrons are used. This is not a comprehensive list of applications, but I've tried to show a broad range of different areas where synchrotrons are used. We won't be able to look at all of these areas today, but we'll look at examples from many of them, including pharmaceuticals, batteries, minerals, and agriculture as we go. The CLS is the only synchrotron facility in Canada. We've been operating since 2005, but many of our beamlines were built more recently or have received recent upgrades. We have about 250 to 300 employees, ranging from scientists and engineers and technicians to finance and human resource specialists. And industry access is a core mandate of the CLS, and it's been a priority since the conception of the facility. Personally, I work in the Industry Services Group at the CLS. We predominantly work with companies, but we also help academics to access the CLS for fee-for-service work. And one of the privileges of our group is we get to work with numerous different beamlines and techniques and look at a wide range of different types of samples. We're very fortunate to get experience in many areas of research, and we're a very good resource if you're looking to discuss different ways of accessing the CLS. The CLS is a relatively small synchrotron facility, but we have a wide range of techniques. Everything from the far infrared region to very hard X-rays that get used for scattering and computed tomography. Generally, as you go around the ring counterclockwise, starting at our far IR beamline, the energy increases from the milli-electron volt range to over 100 kilo-electron volts at beamit. I'm not going to go into detailed technical specifications for individual beamlines. We can end up being here all day, but as I go through examples, I'll try to highlight some of the beamlines that are being used. If you want to better familiarize with our beamlines and techniques, we have resources on the web that illustrate the energy ranges of our beamlines. And each of these individual beamline bars leads to a web page describing the specifications and techniques that are available for each beamline. And there's also a solutions email on this page. So if you've got a specific project in mind, but need advice on how to get started, you can email us and your questions will get directed to someone who's appropriate to answer them. There's three main categories of synchrotron techniques, including diffraction, spectroscopy, and imaging. And I'm going to highlight a range of applications from each of these three categories. Synchrotron techniques in general tend to be very fast. They're element specific with higher resolution and lower noise than laboratory equivalents. And this makes them very well suited to time-resolved studies. Many of these advantages of the synchrotron also lend themselves to hybrid techniques, which blur the lines between these three categories. So I've tried to create some relatively short examples. Some of them are industry-based and some drawing on peer-reviewed research to highlight some of the things we do. And we'll start with some diffraction and scattering experiments. Here's what a single crystal diffraction experiment looks like. The crystal is mounted on a metal base like this, that's roughly an inch in height.
Hey, just curious how she's doing.
To actually see the crystal, we need to zoom in on the top of the base. The crystal is the small cube in the center of the loop. This crystal is on the order of 100 micrometers in size, so it's not very big. When we shine X-rays on the crystal, we get a pattern of spots on our detector. This is a cartoon, but an actual pattern looks something like this. And the pattern depends on the orientation of the crystal. So we collect patterns of spots at many different crystal orientations by rotating the crystal in small steps and collecting a new pattern at each step. Typically, we collect hundreds of 2D patterns from a single crystal. Computers are then used to analyze the 2D patterns and determine how the atoms are arranged in the crystal. Here's what a synchrotron MX data collection actually looks like. This is a set of diffraction patterns obtained from a protein crystal, which typically have thousands of atoms in the unit cell making up the crystal. Synchrotron data collection is extremely fast. On our CMCF ID beamline at the CLS, an entire data set with hundreds of images can be collected in less than 30 seconds. The whole cycle, including mounting and dismounting the crystal using a robot, takes a couple of minutes. You can also operate these beamlines from home. Over 90% of our CMCF users operate the beamlines remotely. The users ship us the samples, we load the samples for their beam time, and the user takes over remotely using a robot to mount the samples. We've seen what the data looks like. Here's a protein structure determined with synchrotron data. A single view of this protein molecule doesn't really do it justice, but rotating it gives you a better idea of the full 3D geometry. This molecule is a human protein complex from a study on the design of anti-cancer drugs. The entire molecule is made up of over 10,000 atoms, and solving a structure like this would be very difficult without the synchrotron. My colleagues, Dennis and Kurt, run huge volumes of commercial protein samples on both of our CMCF beamlines. This work is a critical component of industry revenue. In any given year, the majority of our revenue comes from MX. This work also has some really high impact publications. Shown here is the COVID main protease, sometimes called the spike protein, complexed with a small molecule called GC376. And GC376 is an antiviral medication that's proven effective for multiple human and animal viruses. And it's now being tested as a potential treatment for COVID. The primary purpose of our CMCF beamlines is protein crystallography, but CMCF BM is very good for small molecule, single crystal XRD. My colleague Dennis does protein data collection primarily, but he is an expert small molecule crystallographer as well. Most data sets can be collected in a matter of minutes, larger structures can be solved and better resolution can be obtained than with lab XRD. And you can also use smaller crystals. Dentists routinely solve structures using 20 to 30 micron crystals, and sometimes even smaller crystals can be used for optimal materials. Since you can do powder diffraction with lab sources, it's worth quickly reviewing why we want to use the synchrotron for powder diffraction. Many people immediately think of high resolution because thinner peaks can be very important for indexing and solving new crystal structures, particularly for complex structures. High intensity is also a huge advantage because data collection is faster, you can study much smaller samples, and it's very helpful for in situ and operando studies. Synchrotrons really shine for tracking time-dependent processes. This figure to the bottom right illustrates an in situ study of clathrate hydrate formation at both low temperature and under vacuum. And the combination of high intensity and excellent signal-to-noise estimates from large 2D detectors really improves our detection and our quantification limits, and our ability to get quality data from challenging samples. This point about the 2D detectors is worth discussing in a little more detail. So samples from ideal powders with micron-sized crystals and no texture yield perfect diffraction rings, which are completely uniform. But often this isn't what we encounter from real samples, and particularly with mineralogical samples. Real samples are often heterogeneous, multi-phase materials with a wide range of crystallite sizes, even when they've been extensively ground. But when you integrate the entire 2D pattern, you effectively average the entire ring to partially mitigate that graininess. And combining a large 2D detector with capillary rotation during data collection is a very effective way to get reasonable data for challenging samples. You can see here that for a point detector or a strip detector, both of which are really common on lab instruments, you only see a small portion of the 2D pattern. So getting reproducible peak intensities from grainy samples can be a real challenge. Tunable energy and high energy are also very important. Our Brockhaus beamlines for diffraction and scattering offer a very large energy range that facilitates pair distribution function, or PDF analysis. And PDF is a total scattering technique that can study local structure, even in poorly crystalline and amorphous materials. High energy is also really important for transmission measurements on highly absorbing samples, like metal alloys or battery systems. Structure solution from powders will always be important because many real materials cannot be prepared as single crystals. This powder pattern was obtained from a new organogel material that could potentially be used for applications like drug delivery. And it was collected using the high-resolution end station on our Brockhaus low-energy Wiggler beamline. This material is kind of like an advertisement for using the synchrotron. It has one of the more unusual cells I've encountered. You can see the short axis is less than 5 angstroms long, but the long axis is over 50 angstroms. So properly indexing this pattern was tricky, and the resolution provided by the synchrotron was essential. There's also 2 molecules in the asymmetric unit. I hesitate to say that you couldn't solve this structure with lab data, but it was hard enough using synchrotron data that I wouldn't want to have to use lab data. And given the complexity of the structure, we used density functional theory to help us verify that the structure was correct. And DFT is an excellent tool for structure validation of powder structures. It's also great for precisely locating the light atoms, like hydrogen, that don't scatter X-rays strongly. And the agreement here between the DFT and the Rietveld results are excellent, and they strongly suggest that our powder structure is correct. The ICDD has a long-standing project devoted to determining unknown high-volume pharmaceutical crystal structures. And recently, they've been using the high-resolution powder end station on our Brockhaus low-energy Wiggler beamline for some of these structures. These are some, but not all, of the pharmaceutical crystal structures solved by Jim Kadick and the ICDD team using CLS data. Didn't have room here for the references, but all of these crystal structures were published in Powder Diffraction Journal in 2025. Powder diffraction is critical for looking at new battery materials. This is a recent study of an all-in-one cathode material for all solid-state batteries conducted on the CLS Brockhaus high-energy Wiggler beamline. Lithium battery materials are often really challenging to study because lithium is very light and it doesn't scatter X-rays strongly. So when you have lithium in the presence of heavier elements, you generally need a combination of techniques to get the full picture of the structure. In this case, we've combined the synchrotron diffraction with neutron diffraction, PDF analysis, and ab initio molecular dynamics calculations. And these illustrate the channels through the material that enable the lithium transport. Here's one last structure example. This is an example of a lithium superionic conductor suitable for use as an electrolyte in all solid-state batteries. This material has very high ionic conductivity, on the order of millisieverts per centimeter. Similar to the previous example, the structure was challenging and it required multiple techniques to get the full picture. The cell is fairly large, and this made the synchrotron ideal for indexing the cell and solving the basic columnar structure. But the mixture of very heavy tantalum and very light lithium atoms meant that neutron diffraction with ab initio molecular dynamics calculations were required to help determine the lithium ion positions. And this material works so well because it contains channels where it's very easy for the lithium to move. Along the A-axis, the channels form a continuous pathway of tetrahedral coordination environments with a very low energy barrier for lithium migration. This material will never be used in real batteries because tantalum is too expensive, but the hope is that using mixed oxide halide design of this material to develop materials with similar performance, but using cheaper metals. In situ and operando studies are key capabilities of synchrotrons. And fast data collection can be important for operando studies of batteries, but high energy is also very important because low energy X-rays don't travel far in dense materials. With most lab XRD systems, you can't perform operando measurement in transmission. You have to use reflection geometry because lab XRDs aren't sufficiently penetrating to go through battery systems. But with synchrotron X-rays at high energy, you can perform transmission measurements on real battery systems, like the pouch cell, which we've shown here. And due to the speed with which you can collect data, in addition to operando measurements with the synchrotron, you can actually spatially map different locations as well. A lot of operando studies look at a single location and assume that it is representative of the whole battery or the entire material, but that's often not the case. This is some recent work led by my industrial colleague, Toby Bond, who also completed his PhD with Jeff Don at Dalhousie last year. And these are single peaks from a series of locations in the cathode of a new battery on the left and an aged battery on the right. And the peaks from each location line up really well in the new battery for both the fully charged and fully discharged states. But there's major discrepancies between the locations in the aged battery. Here are 5 locations in the new battery, and you can see that the behavior during charging is virtually identical at each location. The peak widths and shifts are uniform across locations for both the anode and the cathode peaks that are illustrated here. But if you look at the profiles for the aged battery, there's significant differences between different locations. You no longer get complete dilithiation during charging in some locations. Parts of the cathode are either inactive or they've been kinetically hindered. And there's also various degrees of staging observed in the graphite anode of the aged battery. The scale of these differences across the battery are pretty significant. And we will actually come back to these batteries a little later in this presentation. We look at a lot of mineralogical samples in the industry group. With powder diffraction, some of the most challenging samples to work with are clays. This is an example of bentonite, which is a mineral mixture predominantly composed of a turbostratically disordered smectite clay. So instead of perfectly ordered layers in this clay, individual layers are translated and they're rotated with respect to one another. And this gives rise to the broad, really asymmetric features of the powder pattern, which are really challenging to properly model in a physically plausible way. These clays are also very prone to preferred orientation, while the coarse minerals like quartz are very prone to graininess. Here's a Rietveld refinement of this bentonite data. And with the capillary geometry and sample spinning and a very large 2D detector, we can minimize the preferred orientation of the clay and the graininess that's inherent in the coarser minerals like the quartz. And this Rietveld refinement employs the programs Profex and BGMN. And they have customizable scripting, which can be used to build physical models capable of describing really complex phenomena like turbostratic disorder. So pair distribution function, or PDF analysis, is a total scattering technique, and it's a very powerful way for studying poorly crystalline materials. Some minerals have been noted for decades without complete structural characterization because of poor crystallinity or disorder. The mineral arsenosiderite is frequently observed in ore deposits and mine waste, and understanding its structure has important implications for dealing with arsenic in tailings. This work determined the structure of poorly crystalline arsenosiderite using reverse Monte Carlo modeling of the PDF data. And arsenosiderite has a layered structure, again, where shifting between those layers disrupts that long-range order, and it makes it very challenging to characterize. High-energy synchrotron diffraction can be a very powerful tool for strain measurement. With the high-energy wiggler at our Brockhouse beamline, we can do strain measurement on metal alloy samples. This is a welded sample of zirconium alloy, which is a metal commonly used in the nuclear industry because of its corrosion resistance. And zirconium is a quite dense metal, but with 70 kiloelectron volt X-rays, we can easily do transmission measurements through this section. So the client was interested in what is going on in the weld region where the two pieces join here and the heat affected zone that's adjacent to the weld region. There were two objectives for this project. One was to map the phase composition in the weld zone and the heat affected zone. And we can do this by integrating the full 2D patterns and performing Rietveld refinement. The second was to map the residual strain in the weld zone and the heat affected zone. And to do this, we can look at the shape and the distortion of the 2D Debye rings to extract some strain tensor elements. I'm going to focus on the second objective here, the strain determination, which was made using the Debye rings from the main alpha zirconium phase in this alloy. Using a single 2D ring pattern, we can determine three strain tensors. The tensile strain tensors that are parallel and perpendicular to the weld, and one shear strain tensor. In the weld zone at the top right, the strains perpendicular to the weld, the E22 strain tensor shown in blue, are all compressive. The strains parallel the weld, the E11 strain tensor in green, are small and compressive in the middle of the weld zone, but they change to tensile towards the edges of the weld zone. And the magnitudes of the strains tend to be similar in the heat affected zone, or more similar in the heat affected zone than in the weld zone. And the parallel and the perpendicular strain components are more comparable. You can generate these strain estimates from any 2D ring in the pattern. So we chose 8 different diffraction rings from the main alpha zirconium phase to estimate the residual strain tensors for comparison here. We'll move on now to our second category, specifically X-ray absorption spectroscopy. X-ray absorption spectroscopy is a core technique that is mainly performed at synchrotrons because tunable high-intensity sources are really hard to replicate in the lab. XAS is element-specific. It can be used on any state of matter, and it can probe very dilute concentrations, sometimes on the order of parts per million. The X-ray absorption spectrum is normally divided into two components, the near-edge portion, or the XANES, which is shown in red at the top right, and the fine structure above the edge, or the XAFs, which is shown in blue. We tend to predominantly use the XANES in the industry group, and these examples will focus on XANES. And the schematic here at the bottom right represents a typical setup on a hard X-ray beamline, where measurements are undertaken in air. For concentrated samples, direct transmission measurements are made with ion chambers on either side of the sample. For really dilute samples, the XAS spectra are collected using fluorescence from the absorbing element. So here's a hard XAS example. We conducted a recent large industry project looking at fine particulate matter, or PM 2.5, on the Toronto subway system. Subway commuters are exposed to high particulate matter levels, much higher than you'd normally encounter in other locations. Major contributors to particulate matter are actually components of the trains themselves. The mechanical friction inherent in the subway system creates a lot of dust. So Health Canada wanted to understand the sources and composition of particulate matter in the subway with an eye towards better ways to improve the air quality. Air filters were used to obtain particulate matter from 22 different subway stations on both the Young University and the Bloor-Danforth subway lines, plus 2 outdoor sites for reference. The samples were initially examined with X-ray fluorescence to determine the main element species present in the particulate matter. So 6 transition elements were chosen for chemical speciation using XANES, based on abundance and the heterogeneity of toxicity across the species, different species of the elements. 2 elements were of particular interest, though. Iron was by far the most abundant metal species because it's ubiquitous in so many train components. And chromium was also of interest due to the toxicity of chromium-6. So chromium-6 is associated with numerous health issues. In particular, chronic inhalation exposure can lead to lung cancer. Chromium-6 is a very favorable oxidation state to detect with XANES because it has a prominent pre-etch feature. For this element, the analysis was simply a matter of fingerprinting. Can we see the chromium-6 pre-etch feature in the subway samples? Fortunately, there was no evidence for measurable quantities of chromium-6 in any of the subway particulate matter samples. For iron, quantitative XANES using linear combination fitting was required to determine the relative proportions of the various iron species. Magnetite, which is a mixed valent spinel oxide, was the main observed component, with a variety of common iron oxides and hydroxides observed in smaller quantities. And some of these XANES fits were challenging because the iron reference standards are highly crystalline, while the particulate matter samples were poorly crystalline or amorphous. You can see this broad post-edge feature illustrated here with the red circle. And it's so broad because of the poor crystallinity of the iron species in here. So when you combine the X-ray fluorescence elemental quantification with the speciation provided by XANES, you can obtain a fairly complete picture of the composition of the fine particulate matter. All of the main components in the left pie chart here are iron-bearing phases, but the other component is broken down by the pie chart on the right. There's also an unknown component here of unidentified material. But they were able to compare both subway lines. Line 1 shown here is the Young University line, and you can see that the dominant component is magnetite. They were even able to use a chemical mass balance analysis to estimate the contribution from individual sources to the fine particulate matter on both subway lines. This is a second example, and this is a soft XAS materials application. High entropy oxides are emerging materials for applications like energy storage and catalysis. They are interesting materials because they incorporate multiple metals on individual cation sites, which can enhance their stability and also allow you to tune their physical properties. These specific HEOs were prepared with a cubic spinel structure with two cation sites, an octahedral site and a tetrahedral site. And they were synthesized with 5 transition metals plus varying amounts of non-metallic or non-magnetic rather gallium. And you can see based on the EDS maps at the bottom here, the metals are all uniformly dispersed in the materials. What's interesting about this system is that you can tune the magnetic properties by controlling the amount of gallium in the material. You can see significant changes in the magnetic susceptibility and the magnetization curves here as a function of the gallium content. But the key question for understanding the properties is, how are the various metals here distributed across the two cation sites? When you've got two cation sites but 5 or 6 metals, you can't determine where the individual elements are located with diffraction techniques. There's too many permutations, so you need additional information to determine which sites the metals occupy. And soft X-ray absorption spectroscopy is a really good way to look at these metals. You can probe each of the individual elements with soft X-ray absorption spectroscopy to determine their local environment. In this case, they used both X-ray absorption spectroscopy and a related technique called X-ray magnetic circular dichromism, which is an XAS variation using circularly polarized X-rays that are sensitive to the crystal field. There's very little change in the chromium and the nickel in these samples. They both reside exclusively on the octahedral site, but there are significant changes in the site occupations of the cobalt and the manganese. And adding gallium requires charge compensation, which is provided predominantly by the manganese. This work was performed on our Reichs beamline. And in addition to X-ray absorption, Reichs can perform X-ray emission spectroscopy plus resonant diffraction and reflection. It's a very powerful beamline for condensed matter studies on systems like superconductors and magnetic materials. The last category we'll look at is imaging. This is a nice category to end on because it gives some nice pictures. So the first technique we will look at is X-ray fluorescence mapping. Hopefully everyone is familiar with X-ray fluorescence, where we excite atoms with incident X-rays, and they emit characteristic X-rays with X-ray fluorescence that allow us to identify the individual atoms within the sample. The beauty of the synchrotron is the available intensity is so high that we can focus the beam down to a small spot, and we can raster the sample in the beam to produce a map of the elemental composition, something like this. This is a 2D map of the iron and zinc contents in the root nodules of a soybean plant. These nodules have vasculature on the outside, almost like veins. So you can see the iron in red is pretty evenly distributed throughout the nodule, but the zinc in green is mainly in the vasculature system. XRF mapping is excellent for looking at distributions of nutrients or toxic elements in plants. This is a study looking at the distribution of nutrients in oats. If you don't know what these features of the oats are on the top right, don't worry, I don't either. There's a couple of ways this data can be illustrated. You can make individual maps for each element by themselves, as I've illustrated on the left here. But often we want to see how different elements are associated with one another on a single map. This is what's called a tricolor plot with 3 different elements in one map. You can see here the manganese and oxide, or the manganese and zinc rather, form layers on the outside of the oat, while the sulfur is spread throughout the middle of the oat. And the embryo at the bottom right contains an awful lot of zinc. Elemental mapping can be very powerful for understanding the effects of toxic elements on animals as well. Mercury is one of the most widespread toxic pollutants to living organisms, and exposure to mercury is extremely common. Small fish called zebrafish get used for toxicology studies because they have physiology that's actually kind of similar to mammals. So they're a decent model for how the toxins might behave in us. There are multiple elemental maps, including mercury for a zebrafish here, and you can see immediately that the elements are not evenly distributed. If we zoom in on the eyes and the brain, you can see mercury is highly concentrated in the eyes. If we zoom in further still, the mercury appears to concentrate in a layer underneath the cornea and the epithelium. And understanding how elements like mercury distribute themselves in the body can be really important for understanding their toxic effects. Here's another technique called spectromicroscopy. This is definitely a hybrid technique combining spectroscopy and imaging. There are numerous different forms of X-ray spectromicroscopy. The schematic on the right here illustrates a technique called scanning tunneling X-ray microscopy, or STXM. STXM is a soft X-ray technique with very high spatial resolution using a spot size of roughly 30 nanometers. And it requires very thin cross-sections of material on the order of 100 nanometers thick. The sample is rastered in the beam to produce a 2D map while collecting X-ray absorption data at every single step as well. So the result is an image where every pixel contains X-ray absorption data. So the figure at the bottom right illustrates the form of nitrogen in doped carbon nanotubes, where the green represents nitrogen that's incorporated in the nanotube structure. The red is nitrogen gas within the nanotubes. You can perform STXM chemical mapping in 2 dimensions, but you can also perform chemical mapping in 3 dimensions by rotating the sample. So this is done by creating a 4-dimensional data collection stack by rastering in the X and Y dimensions, rotating the sample, and collecting and scanning the energy at every single point. This creates huge amounts of data, and it is computationally intensive, but you can tomographically reconstruct nanoscale objects in 3D. This is a sulfur bacterium, about 1 to 2 microns long, with chemical imaging illustrating the distributions of calcium and carbon and sulfur in the bacterium. By replacing a point detector with a CCD detector behind the sample to collect 2D scattering data, a modified STXM ptychography imaging technique can be obtained. And STXM ptychography further improves the possible spatial resolution down to potentially 1 to 2 nanometers, which is getting very close to TEM resolution. This technique also creates huge amounts of data because now every point on your sample has a CCD image associated with it, and it's very computationally intensive. Here's a comparison of STXM ptychography with transmission electron microscopy, employing an EDS detector to examine elemental distributions. And this work looks at lithium manganese nickel oxide cathode materials for batteries. The STXM ptychography spatial resolution is comparable to low resolution transmission electron microscopy, but the chemical imaging is actually superior because you can image chemical species in addition to just elements. And you can only get the elemental information from the TEM EDS. The last imaging technique we'll examine is computed tomography, or CT. If you consider a familiar hospital CT system where the patient is held stationary and the radiation source moves around the patient, with the synchrotron, we can't move the source. So the sample is rotated in the beam. And similar to other techniques, synchrotron CT is much faster than lab CT, and this makes it very useful for time-resolved experiments. And tunable energy means you can generally get better contrast than lab CT, and you can image much larger areas and image at higher resolution. CT can be very powerful for looking at batteries and the damage that is caused by cycling. Here's an example of a battery pouch cell examined with CT. This is the same type of pouch cell we looked at with XRD earlier. And with CT, you obtain complete 3D renderings of the batteries, which you can section as needed in order to answer your questions. Synchrotron CT is versatile because with control of the energy and the type of detector you use, you can design the experiment to probe different length scales in order to answer different questions. So the far left figures illustrate lower resolution experiments at the battery cell level. At the top left, we have a new battery with limited voids in the electrolyte fluid. But at the bottom left, you can see major depletion of the electrolyte after cycling the battery for a couple of years, and the growth of gas voids. And these gas voids can cause a battery to fail. If we zoom in a little bit, the two middle columns illustrate the damage that cycling causes at the electrode level, like thickening of the electrode layers and formation of voids or delamination of electrode layers. On the far right, even higher resolution measurements illustrate differences in particle morphology and microcracking of individual particles within the electron layers after cycling. A lot of the particles in the cycled battery at the bottom right look crushed or pulverized from the continuous expansion and contraction that happens during cycling. When you combine the CT and the PXRD results that we talked about previously, you start to get a more complete picture of what happens during battery aging, and you can see trends and differences between batteries. Here's a comparison of two aged batteries. A conventional EV battery cycled to the equivalent of driving 600,000 miles, and a battery with single crystal cathode particles cycled to 5 million miles. The conventional EV battery shows extensive evidence of cracking and swelling in the cathode particles. But when we look at the single crystal cathode particles, there's almost no swelling observed. They look almost pristine after all the cycling. For the conventional EV battery, most of the electrolyte has been depleted during the cycling. But if we look at the single crystal cell, there's still plenty of electrolyte left. It's certainly been depleted somewhat, and there are some voids there, but we do have some electrolyte left in the single crystal cell. Finally, we can see major differences with location in the XRD patterns during charging of the conventional EV battery. But almost all of the locations in the single crystal cathode still look homogeneous after cycling to 5 million miles. Synchrotron CT is very fast, so it's much better for time-resolved studies than lab CT. This is an example illustrating changes in the liquid electrolyte of a battery where the electrode expansion during cycling causes the motion in the electrolyte. And the individual CT scans making up this movie take less than a minute. So you can study other types of transient phenomena as well that occur on a similar timescale. You can look at things like liquid penetration in rock cores, microfluidic cells. Really, the sky is the limit, provided the time scale is appropriate to track it. You can use CT to quantitatively map heavy elements using a technique called K-edge subtraction. So what you do is you take CT data sets above and below the absorption edge, and you subtract them to specifically highlight the element of interest. In this case, the researchers were looking at strontium localization at the growth plates of bones, specifically the tibia. And strontium is a good tracer element for new bone deposition. You can see in the juvenile rat, shown here, strontium is widely distributed around the bone. It appears almost everywhere in the bone. In the mature rat bone, shown here, strontium is concentrated around the growth plate, and it's not nearly as widely distributed. I can definitely commiserate with this mature rat. He is probably very sore getting up the next morning after a hockey game. This is the last example of the day. These researchers were studying differences in plant vascular systems in different members of the Astraceae flowers, commonly known as the daisy family. This is a fly-through video looking at the head of a Gerber daisy and illustrating the internal structure of the plant, which was determined with computed tomography. I can't personally do justice to the various features of this plant with words, so I'm just going to let you watch from here. So hopefully I've convinced you that synchrotrons in general and the CLS specifically have a wide variety of uses for both industry and fundamental research. They're particularly useful for challenging problems like heterogeneous materials, very small samples, in situ studies, and really dilute species that are difficult to study with lab techniques. I have to emphasize, this isn't a complete list of things you can do with a synchrotron, just an illustration of some of the possibilities. And we have a lot of experience in the industry group with different techniques around the CLS. And we are always happy to discuss possible projects or the best way for you to address a problem or to access a specific technique at the synchrotron. I would really like to thank many colleagues at the CLS who've contributed to this work, and also some of our clients and collaborators whose data appears here. And with that, I would really like to thank you for your time, and I'd be happy to take any questions.
Please remember to visit icdd.com for information on free webinars, educational events, PDF databases, and JADE software.